Hey everyone, thanks for tuning in. So for those of you who don't know me, I am Ted O'Neill. I'm the president of the Davis Alt.Pro project. And so if this is the first of our events that you're attending or you just want to learn more about our club and just stay in the loop, I'm going to just share our Aggie Life page in the chat. And this is where you can sign up for our email list and just find some of our other contact info if you're interested. So anyway, we do have Mariah Murera here as well, who I think is going to help moderate tonight's discussion. And of course, we are very fortunate to be joined tonight by Dr. Lauran Madden, who is the Vice President of Research and Product Development at BlueNalu. I'm gonna let Lauran give a more detailed introduction of herself, but briefly, she has a PhD in bioengineering from the University of Washington and has held a postdoc position at Duke University. And so she has a lot of multidisciplinary experiences that relate to tissue engineering and 3D cell culture-based products like cultivated seafood. So we're very excited to hear more about her journey and her previous work, as well as how she's helping to accomplish the goals of BlueNalu. And so tonight, I think we'll just let Lauran present the slides and talk that she has prepared for this meeting. And then afterwards, we can focus on questions from the audience and try to have a bit more of an interactive discussion. So audience members can feel free to just type questions into the chat during the talk, and then we can try to address them afterwards. And then also afterwards, you can just, I guess, raise your hand or something to ask a question by voice. So Mariah, are you able to just like monitor the chat for this?
Yes, I'll keep monitoring. Yeah.
Cool. Yeah, so I think we'll just let Lauran take it from here. Thanks so much for joining us.
Hey, thanks, Sid, and thanks, Mariah, for reaching out and setting this up for us. So I'll share, I guess before I share my screen, just a brief background. So as Ted mentioned, I've originally started my career in more tissue engineering and regenerative medicine up until 3 years ago when I joined BlueNalu. So it's definitely been a big shift. So I think I'm going to talk a little bit on, what is applicable to that field, but then also how we incorporate different technologies or look at the different, safety regulations or different requirements of a food product. And so hopefully, as all of you continue your education or doing research, either it'll spark some new areas of interest, or hopefully you find some valuable corollaries to your own educational path. Okay, so can everyone see these slides?
Yes.
Great. So I basically call this, from cells to seafood. And I'm going to give some broad overview, but also some specifics about, kind of the very different sections of the technology development and commercialization of like cell-based seafood products. But I kind of wanted to start out with BlueNalu as a company in general. So when the company first started about 3 years ago, there really weren't any other or many other companies looking at seafood products. So we really sought to be, the leader in the manufacturing of these new class of seafood products. And we definitely feel like this technology can provide, not only great tasting products, but products that are healthier for people, humane for sea life, and sustainable for the planet. And we'll go through a little bit of, some of those benefits in the follow-up slides. But these are produced directly from fish cells and without, any of the environmental contaminants such as, mercury, PCBs, et cetera, that can be found in wild caught fish. And I also kind of wanted to just go into some of our strategy where we do consider ourselves a partner, and I'll mention in later slides, but we actually readily partner with a lot of leading food companies. And one really promising about the food industry right now is there's a lot of very large corporations who are actually interested in improving their sustainability and really being able to create this new class of products that meet some of the sustainability and health goals. So going into that a little bit more deeply, I don't know how many of you are familiar with the Sustainable Development Goals, but it is something that's being used a lot more by companies now to kind of chart, the trajectory of what the company is doing and make sure you're aligned with, the overall values of the company. So just a few that, BlueNalu, we definitely keep close to heart. Of course, this life below water as we deal in seafood products, but also, the climate action and trying to create processes that are sustainable, good health and well-being, trying to create safe products, zero hunger. One of the major benefits is being able to, manufacture seafood in areas where maybe there's not an existing supply of seafood currently, and you can actually help contribute to food sustainability. So these are just a few of the, potential benefits of this new and blossoming industry. And we definitely want to keep this in mind as, not only the company, but also the industry as a whole grows. So a couple of the current challenges, and I think, why are we doing seafood specifically? There's quite a lot of, cell cultured products that are being developed now. And a lot of them honestly are in land animals. However, if you look at the supply chain, one really big difference is that, if an animal is on land, it's very easy to count. It's very easy to track. When an animal's in the ocean, it's very difficult to understand the supply. And, over the recent history, there's been dwindling supply of particularly wild caught fishes. And there's been quite a lot of fisheries that have been overfished or have collapsed entirely due to various fishing pressures. So one thing we definitely do in our species selection is to try to choose some of these species, for example, red snapper or bluefin tuna, that would benefit from having an alternate supply chain that's no longer reliant on this naturally occurring supply. And then one of the other major benefits is that, seafood is regarded as a very healthy food, and it's because it has a great nutritional profile. For the most part, you're getting, high protein content, you're getting a lot of different healthy fats, including omega-3s, and you're also getting a lot of different trace minerals like selenium, that sort of thing, that are actually advantageous. However, one of the really big risks right now is environmental contaminants from various pollutants. So for example, mercury is a well-known one, and the FDA has, published guidance several years ago warning of overconsumption of mercury, whereas we may be able to create products that, for example, have such low mercury that you can eat unlimited amounts. So this is, a paradigm shift in what is even possible in terms of the safety of this classification of products. The other big worry, microplastics and other elements are starting to show up in the seafood supply chain. And this type of production method would also just avoid that type of contaminant altogether. So we believe we can definitely create products that, support human health even above and beyond what's currently naturally available in the wild caught supply. Okay. So I'll start with the, kind of one of our really basic slides, but then we'll start diving deeper. I think this audience, I was told a lot would be into food science, so I don't know what everyone's background in cell culture. So hopefully you will follow me through the rest of this. But in general, we start with just sourcing cells from the animal of interest. So for our mahi-mahi product, we source cells directly from a mahi-mahi. So it will be, genetically identical to that source fish. Then those cells, we essentially feed them in these in vitro environments, and they eat the same things that, fish would typically eat. So, fats, vitamins, amino acids, sugars, and we basically grow those up into scale, and then we can concentrate them and kind of turn them back into that really high cell density fish fillet type product. And then once that product is ready, we can then distribute it the same as you would, you know, a packaged, whether that's a vacuum packed frozen fish fillet, poke cubes, or any other type of, you know, seafood product you would typically see in the market. So what does this look like a little bit deeper? There's kind of what I would call 3 main buckets of the technology that need to be developed. So the first is really the cell lines themselves. So that involves first the isolation of the cells. So you need to make sure that, you know, from the fish that you're getting the cells, the correct cell type, everything is controlled, and you want to have a really high quality of this initial source material. Then you want to be able to proliferate those cells. So that's kind of when the cells are growing, you're feeding them, you're making sure they have the correct nutrients. And BlueNalu has chosen to do this without any genetic engineering. So the cells that we use are non-GMO. And then the last step is really taking whatever cells you're growing and then turning them into the cell type of interest. So for example, if you want to create this nice muscle tissue, your cells need to, you know, turn into myocytes and then myofibers and really express all the proteins that you would normally expect to see in a fish fillet. The next step, once you have these stable cell lines, is to then scale up. This, you know, for a small number of words, it's actually very, very complicated. There's a lot of factors that go into scaling up. I don't know how many people have taken a lot of heat and mass transfer classes, but there's a lot of, you know, nutrient availability, shear stress, mixing parameters that one needs to evaluate as they're moving from the small bench scale up to perhaps a large manufacturing scale. Then when you're producing the tissue, you have to recombine all the cells. And that process also, you know, you can kind of think of, we always say it's similar to like dough extrusions or even some of the plant-based extrusions. So there's a lot of different technologies to basically form products into different shapes with different layers, different sizes, that sort of thing. So it's actually more similar to a lot of traditional food downstream processing. And just to know, I think one of the questions we often receive, you know, in the manufacturing, we don't use any antibiotics, and we do have animal component-free medias that we've developed. So that the only animal that should be in the final product is the fish that you want to see there. And then more on the food side, you know, what are the characteristics of the product that the customer is seeking? So this is really in the product development area. And one thing about fish is it is very unique compared to, for example, chicken or beef. You have a different firmness, you have a different flakiness, and there's a very different taste and aroma profiles. And so I'll go through a little bit, but we definitely benchmark all of these and then try to identify, you know, for the different, for example, a mahi-mahi versus a bluefin, what makes that product unique and then what makes that product desirable. And then you try to optimize your product until you get to that point. So I'm going to dive a little bit deeper into each 3 of those areas. So you can actually see some of the scientific aspects behind it. But this is a basic cell line development strategy. So for those of you who are familiar with muscle cell biology, if you take a biopsy of muscle, you'll have these characteristic muscle fibers. And then on the outside of the fibers are what we call satellite cells. And they're actually the cell type that does replicate naturally during development or during a wound healing process. Interesting thing about fish is that many of them have what's called indeterminate growth. So actually many of these cells naturally replicate in fish throughout their entire life cycle. So it actually provides a very robust source of this satellite cell type. And you can see what they look like in 2D culture in the middle picture. Basically, as single cells, under the right media conditions, they'll proliferate readily. And then under the correct differentiation conditions, you can look at this picture of cell function. The cells actually fuse, form these long myofibers, and then express muscle proteins. So the green stain in this is sarcomeric alpha-actinin, which is one of the contractile apparatus proteins that's pretty typical in muscle fibers. But just to note, all the lines, when we term it a cell line, it's actually greater than 50 doublings. And 50 doublings is, was a number, and is termed the Hayflick limit. So typically in mammalian cells, if you get to this, your cells will start senescing and dying. And we've been able to show that we can go well past this number, not only to 100, but even to 200 or more doublings in our cell lines. And then if you look at the stability of the lines on the far side, in the early passages, when you're below the Hayflick limit, you may have some variability. And essentially, as you go into the cell line selection process, what you're looking for is to gain this stability. And that stability is really essential to understanding and predicting your process performance as you move into these scalable environments. So these kind of three metrics in terms of the longevity, stability, and function are ones that we look at all the cell lines, and then we use those metrics to then select out the best cell lines to move forward. A couple of other things that we do look at cell lines, and I think showing this because I think it's maybe underappreciated when people talk about cells. There's actually a lot that goes into really showing that your cell line is robust. So the first, and what I put on the right, is the identity. One common thing in cell culture is cross-contamination, misidentification, and so like any fish product, we actually use DNA barcoding to make sure that the cells that we have are definitely the species that we think they are, and we can confirm this at any point in the production process. So this basically confirms that the cells that we use are indeed the species we say they are. And then the other thing that we do on every cell line is, again, the stability. As I mentioned, we look at growth, but we also look at protein expression and metabolism. So one thing you can imagine that's going to be very important at scale is that not only are you expressing the right proteins you expect, but you have to monitor the metabolism. So kind of like, and I don't know what people's background are in aquaculture feed, but it gets very expensive if you're wasting nutrients that you put into the process. So you definitely want to monitor the metabolism and make sure that the nutrients that you add to the cells are actually being efficiently utilized. And this also cuts down on the waste of the process. So if you match your cell metabolism to your cell feed, you're actually able to create these very efficient feed processes and then reduce the cost of the overall process. And then finally, sterility is probably the most important thing once you go to manufacturing. So all of the cell lines, as I mentioned, we don't use antibiotics in the process. So how do you control for contamination? So what you do is actually, when you make the cell banks, which are the frozen raw materials for the production runs, you actually do a randomized testing on the bank and make sure that it's free from bacteria, fungi, any sort of viruses that may have been a risk in the process, and also mycoplasma, which is kind of like a small organism that can contaminate a lot of cell cultures. But if anyone's very interested on cell line kind of validation techniques, there's quite a few guidance documents from World Health Organization and FDA. They're for the pharmaceutical industry, but similar concepts apply to the raw materials and the cell banks that we use in cell-based foods. So the next step, once we have our cells, we validated the banks, now we have to scale. And I kind of wanted to add this slide because one thing that struck me when I moved into food from pharmaceutical is just the sheer volume that you're going to want to achieve. And being in the metric tons versus when you're producing a pharmaceutical, you may be in the kilos. And so when you look at the outlook for the market as a whole, for example, this one study by A.T. Kearney really looked at, what is these cultured products going to be, and they anticipate, in the next 20 or so years, it may be 35% of the total meat supply. And that is a very, very significant number. So how do we, in 2021, we're at very little supply, then scale up to this amount, which is, hundreds of thousands of metric tons each year. So in a very simplified graph here, kind of just put a little graphic of, what we feel like that looks like. So as I mentioned in the first stage, you are culturing cells. Typically, most people start in a 2D tissue culture flask. And then you might move to like a bench scale bioreactor. In the facility that we're developing now, we're looking at more of like the 200 to 2,000 liter scale. So these are still relatively small scale, but they can produce, hundreds of pounds per week. And then once you have, developed a robust process in kind of that mid scale, you will gain enough data to then start to design your larger scale facilities. And on the right here is a facility that we had done as a conceptual design to show, potentially what it would look like to make around, I think it was 10 million pounds per year. However, you will see, global seafood production is a couple 100 million metric tons a year. So even a facility of this size is still, small compared to the entire seafood market, which is quite massive. But I think one thing you might note about this, on the right-hand side, there's a lot of kind of stainless steel tanks. These may be reminiscent to, large brewery vessels. So think like Anheuser-Busch. That is kind of the type of tank that you would see at that type of facility. And then on the more upper left-hand side is more similar to like a downstream seafood manufacturing. So if are you freezing it and vacuum packing it? Are you grill marking it? Are you batter breading fry it? Those are the things that we talk about in the downstream product. But a couple of considerations just on the scale up side. And these are all areas that, some of them, depending on what area you might go to in a company, will be more or less important to you. One that I always think about is definitely the raw materials and the supply chain. And this has become, particularly with COVID this past year, the raw material supply chain has become a huge issue. And really thinking about, what are these raw materials? Do you have access? How variable are they? Do you have to ship them long distances, or do you have other local sources? Do you have a single sole supplier? And those are all considerations that when you're at this scale, you really need to make sure that the raw materials supply chain is secured. And also, this is one of your highest cost aspects. So this is also one of the areas that gets the most attention in terms of, can we make this lower cost? Can we make it more sustainable? And then the next two are more about process efficiency. So, every step that you have along this entire production line, you can always optimize it. And one thing that we do and others do in the industry is to develop these basically mathematical models where you can put the entire process into modeling software and then really play with the different triggers, understand what about your process is sensitive, and then what kind of changes can you make to improve the efficiency, which typically reduces cost. The next thing is really heat and mass transfer. As I mentioned, why you scale up in a bioreactor, when you get very large, you start having interesting mixing and heat and mass transfer issues. Are you effectively mixing? Are you generating heat? These are things that you can also model mathematically. And luckily, because the pharmaceutical and other industries have been doing this for quite some time, there's some pretty robust models to help you design facilities. And then the last two are more of a maybe bigger picture, but cost reduction, particularly in the cell-based. Everyone's aware of the very expensive burger that came out a couple of years ago. And so everyone in the industry has been very focused on cost reduction. And that is to get to price parity. So if you think of a bluefin tuna, achieving price parity is going to be much easier than achieving price parity on a tilapia fillet. So these are different levels we can think about. And also if you select different products at different cost levels. So if you have a value added product versus a, plain chicken nugget, they're going to be at totally different cost levels. But these are things we definitely think about as we not only look at the process costs, but then also look at the product selection and then how that works into the overall profitability of the system. And then the last is the risk. So I think, one underlooked thing when you're doing a facility is you do have to do risk assessments about all of the things coming in, but also all the processes happening. And this kind of rolls up a lot into, for example, like a HACCP program, if you're talking about food safety, but there's also other risks like supply chain or things like that that you need to consider and then work into your overall business plan. So one thing that we're doing, just to share because it's very exciting and takes up a lot of my time right now, we're actually building out this 40,000 square foot facility that's hygienically designed GMP facility. Our first product, we're currently scaling up the Mahi Mahi product, but we also have in the pipeline, bluefin tuna, red snapper, and then hopefully this facility will also be flexible enough to produce different types of products. So it's more of kind of like a pilot facility where we're going to test out different processes, different product types, and then be able to do a lot of, kind of limited time offerings or other small product launches to really start to understand the market acceptance and the, flexibility of this technology. But this will be our new home starting later this year. And I also, at the new facility where we're going to be doing GMP manufacturing, I also like to just mention that while we have a slightly different technology, it also is regulated as a food. So there's a lot of things that are very similar, but then there's a few things that are very different. So the one thing that's similar is that, we're going to have similar risks to any other food. So biological, chemical, physical hazards, and we can use, HACCP and FSMA regulations to perform a risk assessment, understand the probability, and then put into any of those control points or critical control points along the process. The other changes that maybe people are a little bit less aware of is, if you're in a standard cell culture process, there's a lot of things that are used that are not food grade. So we actually had to spend a long time, number one, identifying those things. For example, antibiotics, phenol red, small molecules, other things that are commonly used in cell culture that are definitely not appropriate for food, and then either replacing them or just eliminating them altogether. The other thing is the allergens. So for those of you who are familiar with cell culture, there's quite a lot of potential allergens. So for example, the albumins, soy lysates or other things that are often components of media, those we assess the components. You actually have to do a risk assessment. And, if any of these are in the process, you will then have to label that product. So you can imagine if we feed, an animal cell with a soy product or a dairy, albumin, you would then have to label it as that allergy. So those are just some considerations that we look at when we look at what kind of is changing as we go from this cell culture process to the food grade process and how that impacts safety as related to a food product. And so once we actually have the product, there's like a car alarm going off in the carport if you hear any weird noises. But now we get to the more, I guess, food related portion, and everyone is always much more interested in the products. So again, one of the kind of interesting things about, manufacturing a seafood product versus harvesting it is that you have a lot more flexibility in creating these sensory characteristics that are actually desired. So one thing I hear frequently and our chef complains about is, particularly in the United States, people don't like fish because it's fishy, which is a little counterintuitive, but if it's, if the fish odor is too strong, which of course can happen if the species has a lot of a certain, odor molecule, or if the fish is a little old, you can develop these different sensory characteristics. So one thing that we've done in collaboration with Griffith Foods is we actually, for each species, create the descriptive analysis tools and the sensory lexicons that go along with the species so that we can understand, what are those characteristics about a certain species. For example, is it, metallic, bitter, whatever the characteristics are. So you can imagine if you have, for example, a whitefish versus a tuna, one may be have more umami, or one may be more metallic. If you go to like crab or lobster, oftentimes those are a little bit sweeter. So really understanding the flavor profile that you're going for. And then being able to, I'm very fond of actual analytics. So, creating them by consumer testing, but also using analytics in the lab. So we definitely use a lot of texture analysis, do a lot of nutritional profiling to understand, for example, there are actually certain amino acids or metabolites that lend themselves to certain flavor characteristics. So we can use those as potential markers. And then some of the tools like the e-nose and the e-tongue that can help you get a read on, how well your product is really matching a conventional product at a molecular level. But we don't go to the consumer testing until we actually have the, the final FDA kind of quality GMP product. So we tend to do most of the analyticals in the early stage until we move to the larger scale manufacturing. And then some of the products that we've developed to date. So a couple of years ago, we did the yellowtail demonstration down here in, sunny San Diego. We were out, had our corporate chef do a demonstration of our first yellowtail product. And this is just an example of, some of the pictures of that product. And, why not only from, a look, feel, taste is important, but also from the ability to use that product in a variety of ways. So fish is interestingly versatile, I would say. So you have, some species, raw or ceviche are the main applications. Some species grilled, pan-seared, maybe you'll put it in a fish soup. And then some you may batter bread fry. So some are much better for fish tacos than others. But depending on the species, we definitely have to understand what are all the applications that a consumer would use them in. And then we try to test all those applications and make sure it holds up. We're definitely very happy with the first product that, we were able to create a product that performed under acidic conditions, under heat, and also could perform raw. And here on the bottom right are some of our happy customers and investors, and also our CEO on the very right-hand side, Lou Cooper House. So that was a big event, and yeah, just I think one of the things as you're creating a project is maybe not to get too narrow on a single product application, because particularly if you're working with chefs, I've definitely found they like to be very creative. So they will always put your product through their paces, so to speak. And then just to close out, I think Mariah asked me, people are always interested in, particularly this group and the Alt.Pro team, what are the opportunities out there? I think it's great that everyone is in a club like this. You're thinking about technology in the future already, and you're taking the time to learn. So that's really exciting. One thing I would suggest is in the continuing education, I think one great aspect about the field now is that universities such as UC Davis, I know University of Washington, where I did my grad school, Cornell, a few of them are starting actual courses specific to this field, which is really good to see. There's a lot of seminars and workshops online very frequently. And then I will also say GFI and New Harvest, which I'm sure you've heard of, both have a lot of educational material. But also I know that GFI has like a careers page as well. So if you're interested in internships or if you're graduating and looking for opportunities, these are also excellent resources to go and to look at, if there's any job posts. I know we've definitely gotten a few people connected through, through counterparts at those companies. And then the other one is BlueNalu actually has an educational platform as well. So it's called eat.blue. It's a little bit confusing. I think you can get to it through our main website, but it's all about just like information. And as it says, celebrating life below water. So they do a lot of posting on different things related to the ocean, but also technologies on ocean health, animal welfare, and all things life below water. And then always definitely also encourage, kind of to also continue living the values. So I know a lot of us here, we always want to strive to reduce that footprint and, promoting sustainable initiatives. So I definitely encourage all of you to, continue to promote, companies or products that kind of align with those values. And then of course, hopefully some of you will join companies like mine in the future and just be part of the next solution. So that's all I had. And if, happy if people have questions now or later on. I also put my contact in case anyone feels like they need to reach out.
Wow, thank you so much. That was so awesome. Yeah, that was a very informative presentation. I feel like we don't always hear a lot of those details from companies like yours, so that was great to hear, especially about like the technical side of things. So yeah, so I think we'll just allow audience members to start coming up with their questions. You can feel free to put them in the chat, or I guess you could raise your hand and we'll try to keep track of like who's trying to speak. While people are doing that, I know there's already one question, but I just had a quick question because I actually do cultivated meat research myself. So a lot of that stuff I'm actually dealing with myself. And so I was wondering, like, is it more challenging or easier to create like a longer lived cell line from fish compared to mammals or other more commonly used model systems?
Yeah, good question. I will say, so I've actually done it in quite a few species. I do think fish biologically have an advantage because they actually grow more consistently for longer periods of time in the development process. And some of them just have there's some studies that that claim there's perhaps differences in the telomeres, but that is actually quite species specific. So you have to kind of look species by species. But I've actually also developed similar lines from human. So it's definitely not impossible in any species.
Okay, good to know. All right, so Raphael had a question. Can you recreate the flavor based on just one cell type, or does it require different cell types? Can you share if you need to add any components to the media to create these flavor molecules?
Yeah, so it depends on the species. So for example, mahi-mahi is very, very lean. So we actually can just use the muscle cells because it actually has less than 1% fat. So almost all the fat is actually in the cell membranes themselves. So you really don't need multiple cell types. However, if you look at, again, bluefin or a salmon, some of those actually have that marbled composition or that layered composition where the cell types contribute both to the texture as much as they do to the taste profiles. So you actually do require some of these differences. So for example, for bluefin, we have 3 cell types that we work with. And so it just depends on the species, how many cell types we derive, and then what the product attributes you're looking for, which ones actually get incorporated into that final product.
Okay, cool. That's very interesting. In terms of the facility emissions, do you know anything about how your company is addressing those concerns potentially?
So I'm presuming you mean like waste or like related to life cycle analysis?
I would think so, yeah.
Okay. Yeah, I think so. That's definitely something, as I mentioned, when we do kind of the long-term technology development, we look at, what kind of waste are you producing? So even if you have a very high feed efficiency, your cells are going to still be producing like lactic acid or other byproducts. And so if you wash those off of the cells, okay, well, what are we doing with that? Can we recycle it in some way? Is there an alternate stream that is now useful for? I don't know if some people have seen, but like one of the tissue engineering companies in North Carolina, and then also Avant, who's doing some fish products. One thing that they're doing is actually taking the extracellular matrix products and creating like beauty products. So rather than having like collagens from, harvested from fish, you can kind of use some of your waste products in the cell culture in this different category. So that's definitely a lot of opportunities. And we've definitely had a lot of internal brainstorming, try to understand how we can create, these really sustainable or waste stream kind of alternate use situations.
Nice, that is very cool. Yeah, I haven't considered that before. So I see we have Payam raised his hand. I just want to ask a question.
Thank you. Thank you for the great presentation. So my question is about the fat part. You said it's mainly coming from the cell membrane, but as you know, the seafood is the important source of omega-3s. And I know the fish in the ocean get it from microalgae. So in the cultured case, I was wondering, would you add a different, like a separate source of it from microalgae, for example, or you rely on the cell membrane phospholipids, or I'm just curious about how would you meet the omega-3 that is very important for seafood products.
Yeah, no, that's a super good question. And just to be clear, yeah, fish do not metabolize omega-3s. Fish derive omega-3 through the supply chain. So similarly, we have to supply the fish with those critical nutrients as well. So yeah, for example, the different algae or other omega-3 sources, there's a few plant sources as well that have reasonably high levels of some of the omega-3s.
So there is no need to differentiate them to adipocytes, right?
So we do have adipocytes, and we can do that. Again, it just depends on what is your final product that you're looking for. So if you want that texture, then yes, you will want to create, fat loaded adipocytes, because those are actually going to more closely replicate kind of the sensory experience.
Okay, thank you very much.
So we have a question related to media costs, and that's actually a big focus of like my research. So I'd be interested to hear your thoughts on this. But basically, could you just talk a little bit more about the costs of your media and like what are the most costly components and like what are you trying to do to lower those costs?
Yeah, that is funny. When we first started, so we used some of the more basic cell culture medias. I think it was like almost $200 a liter. So we've reduced that over 90% in our personal labs, and I need to do another 90%, I'm sure, in the next year. But some of the main costs, I guess, when you first start out, are either the proteins or growth factors. However, one of the great things about the food industry is that they already produce very similar products. So there's a lot of food compatible technologies out there to produce proteins for very, very low cost. So that's definitely one thing when you think about, switching your supply chains. So as you move from pharma to food, there's just, in many cases, there's 1,000-fold cost differential for a lot of these specialty ingredients. So I think that's just one thing that naturally happens as you switch the supply chains. Of course, as you do that, you have to understand the quality requirements of the raw materials and how those impact the cells. So that's definitely something we spend a lot of time on. And we partner with, major raw materials providers to understand those requirements. To some degree, you can, have a slightly less efficient process if you're, saving 5 times. So those are all things as we do the cost modeling that we also take into account. So there is a balance on, price versus efficiency at the end of the day.
Are you trying to like actually replace growth factors completely, or are you just trying to find less expensive sources for them?
For the most part, right now, we remove anything that's not essential. But then for things that are essential, we just make them way cheaper.
Gotcha.
So if you look at a standard cell culture media, there may be 50 to 70 components. If you take out 30 of them, you've already reduced your cost in most circumstances.
Oh, that reminds me, I had a question. So how do you measure the all of the nutrients and components of the media and like how they're being utilized over time? Like, do you have specific methods for each of the different components?
So not each different one. I will say there's a lot of really great service providers, but also different experts in the metabolomics field. So this is something that's been used for quite a while, and actually even in the fermentation. So I wouldn't be surprised if some of your professors have some experience. But basically you can look at what the cells are taking in, so what they're using from the media, what metabolites are being produced, and then understand the metabolic pathways that are really driving the growth. From that, you can derive your different nutritional usages, and then begin to model. And then there's a lot of mathematical modeling, whether that's a DOE design or whether that's doing, randomized designs and then putting it back into a model. I actually, and maybe Kara wants to help me out, but I think one of your professors actually is an expert in this field at UC Davis. So.
Yep, that's actually my lab.
Yeah.
Cool. So there's a question. Basically, fish of varying sizes have different textures based on the size of the flake in the fillet. So is it possible to produce like a mahi fillet with a very large flake size or a very small flake size, or otherwise just customize that texture?
Yeah, for sure. And this is definitely in the extrusion equipment. So kind of similar to if you're, creating a laminated dough product, you can create various thick or thin layers or do a thick then a thin. So there's a lot of flexibility that you can incorporate into that process.
Awesome. So could you talk a little bit more about like which aspects of your production process need to be in the GMP facilities, and how do you think that might change during regulatory approvals?
Yeah, so the GMP portions are from, I kind of mentioned those cells. So part of that validation process is what you go through to do a lot release into GMP. So the starting point of any run is really like a frozen vial of cells. and that frozen vial is tested for many things. I didn't even list all the tests. I think I had in our process over 47 different like unique assays that we run just to ensure, the safety and quality. But once you go through the panel, it's released and then it's fully under GMP. So, controlled storage, controlled access, all the proper controls are in place. And once you thaw that vial, anywhere from that to the final packaging is all under the food GMPs.
Okay. So now there's a question about the nutritional aspects. Have you been able to assess the nutritional values of your products yet? Make sure they're the same as conventional fish? Yes, so we'll start with that question.
Yes, so short answer, yes. But we definitely do a lot of amino acid profiling and fatty acid profiling. Amino acid profiling is pretty consistent in fish. Fatty acid profiling is all over the place, to be quite honest. Seafood products, depending on, there's probably a few fatty acids that are consistent, but depending on their age, whether it's spawn season, are they in South America, are they in Asia, where the fish is caught, you get really wild variability in some of the fatty acid profiles. So we tend to focus a little bit more on the amino acids and then focus in on those, handful of critical fatty acids. For example, the omega-3s or other ones that really give it those characteristic flavor profiles.
Do you need like a lot of product to be able to do all of these nutritional tests, or?
Yes and no. So at small scale, we tend to use like non-AOAC methods. So you'll still do like a single lab validated method. I don't want to go too much into method validation, but there's a lot of devices where you can do amino acid or fatty acid profiling with, very small samples. They tend to be much more expensive. And then once you do your final food product, we work with labs that are validated and use AOAC or other accepted methods for food testing. And typically those require much larger volumes. So we don't do those like necessarily on the bench scale, but we do them on the manufacturing scale.
Okay, cool. So then there's also a question on just like scaffolds. So are there different types of scaffolds that you've had to develop to produce the desired shapes and textures?
Yeah, so BlueNalu actually does not use a scaffolding technology. We use the extrusion technology. I will say in my previous life, where actually in grad school and then postdoc, I did a lot of both synthetic and natural. There's a lot of options out there. So, depending on what kind of architecture you're going for, in some cases that a scaffold may be preferred. So I know quite a few of the meat companies are using it. They also require a much stiffer product. So if you look at a lot of fish are actually maybe less firm than some of the meat products. And so you just have to take into account what type of texture you're actually trying to produce. One of the things that we noticed early on with scaffold is that if you think about how you seed a scaffold and then the length of time it incubates, it actually creates this intrinsic scaling issue. where, you know, if you have 2 or 6 scaffolds, that's fine. If I now have, you know, 200,000, you know, how does that process look? What does it look like? How can you make it efficient? And what happens in scaffold is it essentially becomes a scale out process, which is inherently less scalable than a scale up process.
Okay. Very cool. So if there are no other questions in the chat, or if there's no one else raising their hand, I had a question, just like an overall general question of like, what is the work culture like at BlueNalu? Like, do you enjoy working there? Is it like a
I love working here. I mean, I was actually employee number 1. So I've helped hire most of the people, but also just help the team grow. At any startup, you know, there's a lot of pressure to produce results. So we definitely have a very motivated, focused team, and people do put in a lot of work. However, it's probably one of the most energetic teams I've worked with. And so when you go into the lab, you know, we kind of have a little more relaxed environment than maybe some pharmaceutical companies. Like, we'll play music or we'll have happy hours. One of our office mates has dad jokes that he likes to leave on our desks and just things like that. It's just a very fun environment, and there's this kind of like family feel, just because you spend so many hours together and you're all really working towards the same goal. So I definitely love the culture that we've created.
Great to hear.
So I have a question adding on to Ted. Like, what does your daily work day look like for you at BlueNalu?
Well, I am at the less fun point of my career where I am in lots of meetings every day. So my job right now is more to manage all the technology development activities. So I will have meetings with the upper level scientists, the project teams, but I'll also have meetings with like the executive teams, you know, contract research, you know, potential technology partners, and kind of just understand, you know, where are we right now? But my job is a lot more of where do we need to go and how are we going to get there? So I definitely have to understand, you know, what technologies can we bring in? Who do we need to hire? Do I need to buy another microscope? You know, all these things that will continue to basically allow the staff to do their job. But I guess, you know, when I first started, it was in the lab probably 6 out of 8 hours a day. And that's, I think, what a lot of the folks in the lab experience is. You have, you know, your lab work, you have to do your prep, your documentation, you know, designing experiments, executing experiments, and then preparing, you know, when they meet with me to explain their amazing results.
Great, thank you.
All right. So I don't think there are any other questions. In that case, Lauran, is it okay if students maybe try to just connect with you on LinkedIn or anything like that?
Yep, that's fine.
Cool. Well, yeah, so thank you. Thank you so much. Yeah, this was great. I definitely learned a lot about fish culture and realized that it's somewhat different from what I do with actual chicken cells. So very cool. Yeah, with that, I guess have a great night. Take care, everyone.
Thanks for the
Thank you
invite.
so much.
Great talking to you.